Hey everybody, we are back for part number 2 of our lecture on cells, membranes, and transport. Last time we've been talking about passive transport, which we know is driven by diffusion, and we talked about simple diffusion versus facilitated diffusion. Today we're going to talk about a special type of diffusion, which is osmosis. And osmosis is a diffusion of water through a semi-permeable membrane. So in osmosis, water is diffusing, and it's diffusing through a selectively permeable membrane. So a selectively permeable membrane, for example, would be our cell membrane, which we see with the phospholipid bilayer here. And osmosis, the diffusion of water molecules, these water molecules go through this channel protein in this cell membrane. So osmosis primarily occurs in our cells by what kind of diffusion? Facilitated diffusion. We do have the occasional water molecule doing simple diffusion over here. Remember, the interior of the lipid bilayer is very hydrophobic, so water molecules don't like to be in here. So osmosis primarily occurs by means of a channel protein, and this channel protein is actually a special kind of protein called an aquaporin, like a water passageway, basically. All right, so that's our definition of osmosis. And I want to point out that if we look at this graph, this picture here, I see I have a semi-permeable membrane in the middle, these dashed vertical lines here, semi-permeable membrane. I got water molecules on the left, I got water molecules on the right, and I have more water molecules over on this side, on my left versus on my right. So water molecules will tend to move from this side to that side. So water molecules are going from there's more water to where there's less water, high water concentration to lower water concentration. But notice that water is not the only thing we have here. We also have molecules of sucrose, sugar molecules, on this side. So this side has some particles, some sugar particles besides water. So note that as water goes to where there's a lower water concentration, water is also moving to the region with the higher particle concentration. And that's usually how we describe osmosis. Osmosis goes to where there are more particles, which is just another way of saying where there's less water. Because if there's more particles, well, then there's going to be less water. It's kind of by definition, right? If you're taking up space with particles, there's less space for water. So water diffuses towards where there is a greater particle concentration. And at diffusion of water through a semi-permeable membrane, that is osmosis. All right. So here is a question for you. We got a beaker. We got a watery solution in that beaker. There is a semi-permeable membrane running down the middle here. I have water on both sides. I have sugar molecules on both sides. Water can go through this membrane, but the sugar molecules are too big. So looking at the picture here, which way will osmosis occur? In other words, which way will water diffuse? Will water diffuse from right to left? Or will water diffuse from left to right? Take a moment, think about this. Well, notice, and pause it if you need to really noodle it, if you need to really cogitate on it. And notice that on this right side, I have more of these red sugar molecules. If there's more sugar molecules on the right, well, then there's less water over here. So this right side has less water. So the correct answer here is water is going to diffuse this way. Osmosis is going to happen from left to right because this is where I have more particles. Water goes to where there are more particles. All right, good deal. Let's talk about some different solutions and let's describe how solutions can affect the movement of water into and out of a cell. So there's actually three types of solutions that we have based on whether they make water go into a cell, out of a cell, or neither. And those three possible solutions are isotonic, hypertonic, and hypotonic. Okay, so let's suppose I put a red blood cell, here's a red blood cell, I put it in a solution that's called an isotonic solution. In an isotonic solution, the particle concentration, so the concentration, out here of particles, is the same as the concentration of particles inside the cell. So there is no difference in particle concentration. So that means there's going to be no difference in water concentration. So that means every time a water molecule goes into this cell, a water molecule goes out of this cell, and then there is no net movement of water. So no net movement of water. So in an isotonic solution, there's no net movement of water either into or out of the cell. So the cell is not going to change in size. This little delta here means change. There is no change in cell size. So if I put a cell in an isotonic solution, the solution's concentration and the cell's concentration of particles are the same. No water going in, no water going out. No change in cell size. A lot of intravenous solutions are going to be isotonic because we don't want, cells to change size necessarily. Okay, now let's talk about a solution that is hypertonic. So a hypertonic solution is a solution where the particle concentration is greater than the cell concentration. So now there are more particles outside the cell than inside the cell. So more particles outside than there are inside. So remember, water goes to where there are more particles. Think about particles sucking the water out. So water is going to tend to leave the cell here. Water is going to tend to leave the cell. A hypertonic solution makes water leave the cell. So what's going to happen to the cell? It's going to shrink. So these cells are shriveling up. So the cell gets smaller. And that decrease in cell size is called cronation. So a hypertonic solution has a greater particle concentration than the cells. So it, quote unquote, sucks water out of the cells, and the cells shrink, and that shrinking is cronation. So, isotonic, there was no osmosis, no net movement of water in and out. In a hypertonic solution, the osmosis is happening and water is diffusing out of the cell. And the cell gets smaller, and that shrinkage is called cronation. Okay, there's also hypotonic solutions. In a hypotonic solution, check it out here. There are fewer particles outside the cell, fewer particles in the solution than there are inside the cell. So water is going to enter the cell because this is where I've got more particles. So osmosis is occurring, but water is going into the cell. So in a hypotonic solution, water enters the cell. And this is going to cause the cell to increase in size. And potentially, lysis could happen. Lysis is cellular bursting. Like so much water could come in that the cell explodes. So that's what's going on in a hypotonic solution. The solution has fewer particles, more particles inside the cell, so water is going to go in and the cell is going to get bigger. Worst case scenario, the cell could undergo lysis. It could break open. Fantastic. Those are my 3 ways I can describe solution based on what what direction osmosis happens if we put a cell in that solution. All right, do me a favor. Label these 3 situations. So the blue rectangle represents a solution. We got a red blood cell in each one. Which of these solutions is isotonic? Which is hypertonic? Which is hypotonic? So go ahead, label those for me. Pause the video if you need to. Give you a moment. All right, notice the cell is shrinking here. So this must be the hypertonic solution. There are more particles outside than inside, so water is diffusing out, osmosis is going out, and the cell is shrinking or cronating. In this situation, the cell is getting bigger, so this is going to be my hypotonic solution. Fewer particles outside, more particles inside, so water goes in. Cell is enlarging here. And lastly, I got equal amounts of water going in and out, so the particle concentrations are the same, cell size is staying the same, and this is going to be iso. Hyper means more, hypo means less, iso means the same. So think about a hypertonic solution as having more particles. Hypotonic, less particles, fewer particles. Isotonic, the same number of particles. All right, excellent. Got another one for you to do here. So I got a solution. All right, got a solution here. This little rubber bag represents a cell. There is sugar water in this solution, 0.01 molar sucrose solution. Inside the cell, the concentration of sugar is higher. It's 0.03 molar. Sugar cannot go through the membrane of this bag, but water can. So which way is water going to go? Is water going to enter this cell, or is water going to exit the cell? Think about it for a second. What do you think is going to happen here? Well, did you come up with an answer? All right, let's check your answer here. The cell has more particles. The cell has more particles. There's more particles in here. I'm just drawing particles here. More particles, more sucrose is in here. There are fewer particles out here. So water is going to have a tendency to go in to the cell. Water is going to enter the cell. The cell is going to get bigger. So the solution has less particles. Well, what's the word for that? Less particles, lower particle concentration. So my prefix I'm going to use is hypo. My solution is hypotonic to the cell. Now notice that if the solution is hypotonic to the cell, we can flip this sentence around and we can talk about the cell as being hypertonic to the solution. That says hypertonic. Not the best handwriting on that one. All right, good stuff. Hopefully you got those right, or if not, hopefully you understand what's going on. Okay. Going back for a moment here. Remember, if we are going downhill, that doesn't take any energy on our part, right? We just coast, we roll downhill. That's our analogy for passive transport. But if we're going uphill against our concentration gradient, that is going to be harder. That is going to require energy. All right, so downhill is passive transport. That's down the gradient. Uphill is active transport. That's against the gradient. And there is a type of active transport called primary active transport. That is where our cell is going to take a molecule of ATP, smash it into ADP. So ATP, remember, is adenosine triphosphate. We smash it into adenosine diphosphate and a phosphate. And the energy released is used by a transport protein here. So there's ATP. Protein is smashing ATP, using the energy to move this molecule, this red guy, from an area of low to an area of high concentration. And that's going uphill. So if the protein is directly smashing an ATP to move a molecule against its gradient, that is called primary active transport. Now, a great example of primary active transport in your cells is this protein in your cell membrane. It's called the sodium-potassium pump. It is ubiquitous in your body. Pretty much all your cells have sodium-potassium pumps. Roughly 40% of your calories from food, your energy from food, go towards powering the sodium-potassium pump. Sodium-potassium pump is breaking down a molecule of ATP into ADP and a phosphate, and it's using that energy released to actually move 3 sodium ions out of the cell and move 2 potassium ions into the cell. And both those ionic movements are against their gradient. And so this is going uphill for both these guys. And so this is an example of primary active transport. It is one we're going to encounter again when we get to how nerve cells work and how muscle cells work. So, moving against the gradient, moving uphill, that's active transport. If a protein is moving things against their gradient by breaking down ATP to do so, that is primary active transport. A great example of that is this thing called the sodium-potassium pump. Okay, good stuff, good stuff. Another type of active transport is on a larger scale. It is called vesicular transport. And this is where I'm moving large materials or large amounts of materials or fluids into or out of my cell via these little membranous bags that are called vesicles. Vesicular means involving vesicles. If I'm moving large amounts of materials or fluids out of my cell with vesicles, that is called exocytosis. If I'm moving things into my cells with vesicles, that is called endocytosis. And both of these processes require the cell to spend energy, so they are both examples of active transport. Now here we have some exocytosis. This phospholipid bilayer up here is my cell membrane. So this is inside my cell. This is outside my cell. Here I have a little membrane bag. Notice it is also a phospholipid bilayer. And there are some little green guys in it. And what's going to happen is this membranous bag, the phospholipids in this bag, are going to fuse with the phospholipids of the cell membrane. Remember, the cell membrane is a fluid structure, so we can fuse these two membranes together. And then look what happens as we fuse these two membranes together. These guys here, these phospholipids join up with these guys here, and the membrane has become a little bit bigger. It's got this little down part here. And now the green guys are no longer inside this vesicle. They've now been released outside the cell. And this process is exocytosis. This is a vesicle-based movement of materials, and these could be proteins, they could be hormones, neurotransmitters, even toxins, and we are getting them out of the cell. All right, if we flip it around, we're going to get endocytosis. Again, I got a phospholipid bilayer. This is my cell membrane here. Outside my cell, inside my cell. I've got these purple guys here. Notice how the cell membrane is dipping next to the purple guys. And then what's going to happen is these phospholipids right here are going to move this way. These phospholipids right here are going to move this way. And we're going to join them, and we're going to pinch off a little bag of membrane with the purple guys now inside. And this is a vesicle that we formed by joining the phospholipids here to the guys here. And now the purple things, which were outside of the cell originally, are now inside the cell in a vesicle. And this is called endocytosis. And this process is a vesicle-based active transport of materials such as proteins, iron, vitamins, hormones, and more. But we're going into the cell. Okay, good stuff. There are actually two special types of endocytosis. One is called receptor-mediated endocytosis. The other is called phagocytosis. Receptor-mediated endocytosis is just like the endocytosis I just showed you. But let me backtrack for a second. In this case, the purple guys were just kind of like hanging out right here. The cell membrane didn't do anything to like grab them. But in the case of receptor-mediated endocytosis, there is a receptor, a protein in the cell membrane that grabs onto whatever thing, whatever ligand we want to bring in. So again, we got a little dip in the membrane here. This spot is going to move this way. This spot is going to move that way. The membrane here, the membrane right here and right here are going to fuse together, creating a new membrane with the green guy now inside, but we actually grabbed onto him with a receptor. So this lets my cell engulf specific things. So we use a specific receptor, which is an integral protein, a protein embedded in the cell membrane, and it lets me engulf specific things. Because not, no like random thing is going to grab onto the receptor, but a specific ligand is. And then we make a vesicle and we bring that thing inside. So that is receptor-mediated endocytosis. Very good for bringing specific things inside your cell. The other type of endocytosis is called phagocytosis. It is a special type of endocytosis performed by white blood cells. This is a white blood cell right here. And this is some foreign thing the white blood cell wants to destroy. Maybe it's a pathogen, a dead cell, a broken bit of cell. And these two parts of the white blood cell are called pseudopods. Pseudopods means false feet. The pseudopods stick out right next to the thing to be engulfed. The membrane right here and right here. These two membrane parts are going to kind of like reach out and clasp together. And when we've done that, when these two parts have fused together, then this, let's pretend it's a bacterium, this bacterium has then been engulfed. And that is called phagocytosis. Cellular, cytosis refers to cellular. Phago means eating, so cellular eating. The white blood cells that do this the most are known as macrophages. Macrophages, big eaters. This is a crucial job of our immune system. It's how we kill lots of foreign things. It's also how we do a lot of cleanup when we damage tissues and break cells. Okay, Let's look at one more picture of this. So here we have some phagocytosis going on again. This entire big thing here is my macrophage. There's a bacterium. My macrophage sticks out these two arms, which are called pseudopods. This spot and this spot, they're going to fuse together. Once they fuse together, my bacterium is enclosed in a little vesicle. It's actually called a phagosome, eaten body. And then the phagosome is actually going to fuse with an organelle called a lysosome. There are digestive enzymes in here. And what the enzymes are going to do once these two have fused is they're going to chop up that bacterium, destroying it. And then the white blood cell actually does some exocytosis. It releases the pieces, the leftover, molecular bits of that destroyed bacterium. So that is some endocytosis on one end and some exocytosis on the other end. And with that, we're done with our active transport. And we are going to switch gears a little bit and finish off this part 2 of this lecture by talking about some special ways to join cells together. We know we need to join cells together to make groups of cells, to make collections of cells, in other words, tissues. And we're going to talk about tight junctions, desmosomes, and gap junctions. We already mentioned 2 of these in the lab on epithelial tissue. But let's go ahead and go over them again. So what I've got here are some neighboring epithelial cells. There's their nucleus, some neighboring epithelial cells. I see some microvilli on their apical surface here. And these neighboring cells have tight junctions linking them together. Tight junctions are these special protein complexes that seal neighboring cells together so that materials that are out here cannot then sneak between cells. This is not allowed. So tight junctions seal neighboring cells together to prevent things from sneaking between them. Tight junctions are really important in your small intestine. That's where you do absorption. Up here we got the lumen of the small intestine. I'm going to find nutrients in here, vitamins, water, minerals, but I can also find bacteria and viruses. Here are the cells lining it. There are going to be tight junctions between these cells here. There are going to be tight junctions between cells here. And that's going to prevent any bacteria. Let's make the bacteria. It's going to prevent any bacteria. That's a B for bacteria. We don't want bacteria to be able to sneak through here and get into our underlying tissue, our blood vessels. So tight junctions are going to prevent that. So this is the lining of the small intestine. Cells here are adjoined by tight junctions to prevent things from sneaking in between. Okay. A desmosome is a protein complex between 2 cells. So here's my desmosome. And a desmosome is going to connect these 2 neighboring cells really strongly. So when I want to physically attach neighboring cells together to prevent them from separating, a desmosome is what I want. So tight junctions are good for sealing 2 cells. Desmosomes prevent cells from being ripped apart. Desmosomes are really important in our epidermis of our skin. So this is the epidermis of the skin here. And neighboring skin cells here, skin cell. And these little dots between them are desmosomes. I want my skin cells to be strongly attached to one another so they don't separate, because that would reduce their effectiveness as a protective barrier. So desmosomes are going to strongly anchor skin cells together, and that's really important. All right, my last guy that joins cells together. So I got a cell on the right, I got a cell on the left. Check out their plasma membranes. My last structure is this thing called a gap junction. It is a protein complex that basically forms a double passageway, like a double channel connecting two cells. It's going to let cells communicate and also let cells transfer material between them. So ions and small molecules can flow from one cell to the next through gap junctions. Now, gap junctions are very much found in the muscle of my heart. Here are some cardiac muscle cells. These pink lines are actually where we find the gap junctions. The gap junctions let the heart muscle cells all work together as a coordinated unit. They can communicate with one another via those gap junctions. Okay, and that is it for part 2. I think we were a little quicker in part 2 than part 1. And good job. If you have any questions, of course, come talk to me, let me know. If not, that's great too. Come talk to me anyway. And I will see you next time. Bye-bye.